Welcome to part 2 of the Development and Heredity lecture. In this specific lecture, I will be focusing on heredity and genetics. First, I'll talk about several genetic disorders. Then I'll introduce some basic genetic terminology, and we will work through several simple genetic problems using Punnett squares. This slide introduces some terminology that will help you understand genetic disorders that are caused by an incorrect chromosome number. When an individual has the correct number of chromosomes in each of their cells, we call that condition euploidy. Ploidy is referring to the number of chromosomes. Eu means true. So it means that they have the true number of chromosomes, or in other words, the correct number of chromosomes in their cells. For humans, we have 23 pairs of chromosomes, or in other words, 46 total chromosomes normally within our cells. You get 23 chromosomes from one parent, and you get 23 chromosomes from the other parent to make 46 chromosomes in each cell of your body. However, there are individuals that have an incorrect number of chromosomes resulting from these chromosomes not separating correctly during cell division, or in other words, during meiosis, the development of the sperm and the eggs. And in this case, we call that condition aneuploidy. So aneuploidy would be having an incorrect chromosome number. This makes sense because the prefix "an" means without. So this means without true ploidy, without true chromosome number. There's two main types of aneuploidy. Trisomy is one condition, and this is when an individual has 3 chromosomes of a type instead of 2. And monosomy is when an individual has only 1 chromosome of a type instead of 2. So in a euploidy condition, you should have 23 pairs of chromosomes. So there's usually 2 chromosomes in each pair, right? However, in the trisomy condition, for example, in this case here, you can see that there are 3 chromosomes here of this type when there should only be 2 chromosomes of that type. If somebody is monosomy, they would only have 1 chromosome of a type instead of the 2 that you should have. Not all aneuploid conditions are as detrimental as others. So remember that we have 46 chromosomes, or 23 pairs. So what that means is of each type of chromosome, we have 2 of that type in a normal condition. The first 22 pairs of chromosomes do not determine the sex of the individual. So these are known as non-sex chromosomes, or autosomal chromosomes. So you have 22 pairs of autosomal chromosomes. However, the 23rd pair of chromosomes would determine your sex. If you are XX, then you would be biologically female, and if you were XY, you'd be biologically male. However, one thing I want to point out is that aneuploidy of non-sex chromosomes is usually lethal because these chromosomes have a lot of different genes on them that control a lot of different things in your body. So if you have an extra one of these or you're missing one, usually it ends up in the fetus not being able to survive. The only trisomies that are viable is trisomy 13, which is a condition called Punnett syndrome. Trisomy 18 as well, and then trisomy 21, which is more commonly called Down syndrome. And these are the only 3 trisomies of the autosomal chromosomes that are actually viable. And even with these conditions, a lot of times individuals will not have a very long lifespan. I think with Punnett syndrome, it's only usually a couple years that they may live. However, aneuploidy of sex chromosomes, so the 23rd pair of chromosomes that you have, is a lot easier to survive. So there could be, normally you would have either XX or XY. However, there are individuals that are, for example, XYY, and they can usually live a very normal lifespan, and sometimes there's very minimal side effects to this. That's because these genes on the sex chromosomes mostly control sexual characteristics and don't have a lot of other genes located on those chromosomes. Now let's look at some common aneuploid genetic disorders. The first one that we're going to look at is Down syndrome, also known as trisomy 21. So this is an aneuploid condition caused by an extra autosomal chromosome. And this occurs in individuals that have an extra 21st chromosome. So instead of having 2 chromosomes here, they would have 3. And just by having 1 extra chromosome, these individuals have many physical abnormalities as well as intellectual impairments. Down syndrome occurs in 1 in 1,400 births in women between their 20s and 30s, and the chance of having a child with Down syndrome increases as a woman gets older. So when a woman has a child in her 30s and 40s, the chance of having a child with Down syndrome is 1 in 750 births. The rest of the aneuploid genetic disorders that we're going to be looking at are aneuploid conditions associated with the sex chromosomes. The first one we're going to look at is called Klinefelter syndrome. Klinefelter syndrome affects males because they are born with an extra X chromosome. So normally males are just XY. However, some individuals are born XXY. Having this extra X chromosome can lead to infertility as well as learning disabilities in these individuals. In addition, these individuals will have some female characteristics such as enlarged breast development, wider hips. They may also exhibit smaller testicular size as well. Klinefelter syndrome occurs in 1 in 500 to 1,000 people, so it's actually relatively common. And if you would like to know more about what it's like to live with Klinefelter syndrome, here's a link to a YouTube channel from Ryan Brigante, and he talks about personally having Klinefelter syndrome and the things that he has to deal with that. I will put a link to his YouTube channel in the description below. The next disorder that affects males is called Jacob syndrome, and this is caused by males being born with an extra Y chromosome. So instead of just being XY, they are XYY. Having an extra Y chromosome has very minimal effects and really doesn't affect males as much as having an extra X chromosome. The symptoms are very minimal. Having Jacob syndrome, these individuals may have some learning disabilities, but otherwise they probably don't even know that they have an extra Y chromosome. And this occurs in 1 in 1,000 males. So why does having an extra Y chromosome not really cause many side effects? That's because the Y chromosome is really small, and there really aren't a lot of genes on the Y chromosome other than those that control the development of male sexual characteristics. The next two genetic disorders that we're going to look at are genetic disorders that affect females. Poly-X females are females that are born with an extra X chromosome. So instead of just being XX, they have 3 X chromosomes. The symptoms in these individuals is very minimal, and usually poly-X females, the only symptoms that they may exhibit is that they might be tall and thin. So many poly-X females probably don't even know that they have an extra X chromosome. This occurs in 1 in every 1,500 females. The last condition that I want to introduce to you is called Turner syndrome. Turner syndrome is a monosomy condition because these females are born with only 1 X chromosome. So they are missing that second X chromosome. There are a lot of symptoms associated with only having 1 X chromosome because you have half the genetic information than what you typically would have. These symptoms include short stature, non-functioning ovaries, which leads to infertility, skeletal abnormalities, as well as heart defects. Because of all of these side effects, the life expectancy of somebody with Turner syndrome is only 13 years old, and this occurs in 1 in 2,500 newborn girls. Now I'm going to provide you with an introduction to some very important genetic terminology. You want to have a very good understanding of this terminology in order to solve the genetic problems that we're going to be doing later on in this lecture. The first term that we're going to look at is a chromosome, and this is simply a strand of DNA. And a lot of times a chromosome is depicted like this. It's very simplified, but this is a strand of DNA here, and this is the second strand of DNA here. You have 23 strands of DNA inherited from each parent, so that means you have 23 chromosomes inherited from each parent and 46 chromosomes total in each one of your cells. On these chromosomes, you have sections of DNA that we call genes, and each gene or section of DNA controls a specific trait within your body. An important thing to remember is that you have 2 copies of each gene, and that's because you got 1 copy from one parent and 1 copy of that gene from the other parent. Although you inherit 1 gene from one parent and another gene from another parent, these genes may not be identical to each other, and we call variations of a gene an allele. So for example here, you could see that from each, let's say you inherited this chromosome from one parent and this chromosome from the other parent. Both of those chromosomes have a gene for skin pigmentation. So you have 2 copies of the gene for skin pigmentation. However, one of these copies that you got would actually cause you to have normal skin pigmentation, whereas the other gene that you inherited from the other parent would actually contain the gene for albinism. And so you can see that there's variations in these genes that you could inherit. There are 2 major variations of a gene that can be inherited. One is called a dominant allele, and that is because if you inherit that variation of the gene, it will always be expressed if it's present. So you only need 1 copy of that allele to express that trait. And this is usually, a dominant allele is usually represented by a capital letter. A recessive allele, on the other hand, is not expressed. So this variation of the gene is not expressed when paired with a dominant allele. Therefore, you would need 2 copies of the recessive allele for it to be expressed. And a recessive allele is usually displayed as a lowercase letter. Imagine that this is a chromosome pair in an individual. One of these chromosomes, again, was inherited from one parent. The other one was inherited from the other parent. And let's look at the gene for skin pigmentation. They inherited the dominant allele from one parent and the recessive allele from the other parent. This individual, because the dominant allele causes normal skin pigmentation, what type of skin pigmentation would they have? Well, they would have normal skin pigmentation because by just having one dominant allele, it dominates the recessive allele. It masks the recessive allele. So although they did inherit an allele for albinism, they're not going to display albinism because it's masked by that dominant allele here that's present. It is really important to remember that you're getting one allele from one parent and the other allele from the other parent for a specific gene. And this figure here really demonstrates this well because this is showing my 23andMe results from genetic testing. And I'm half Japanese. My mom is Japanese, my dad is European, and you can actually see that in each chromosome pair that I inherited one of those chromosomes from my mom and one from my dad, because one of the chromosomes is red in color, which is signifying genes that are more common in Asian people, and then you can see that the other chromosome is blue or greenish in each pair, and those chromosomes have genes that are more common in people from Europe. So here you can see a blue chromosome and then a red one in that pair, blue or greenish and red, and so forth. So each pair of chromosomes that I have, you can see that I inherited one from my mom and one from my dad. It's really easy to see this pattern with my genetic background. However, if you were to look at my husband's, it would be much more difficult for him to figure out which chromosome he got from each parent just by looking at his genetic makeup. And that is because he is Puerto Rican, and people of Puerto Rican descent have a very diverse ancestry. So there's a lot of mixing of DNA happening within their chromosomes. And so you can see it's just a very colorful rainbow as you look at each one of his chromosome pairs. Now let's look at some other terminology that's going to be really important in understanding how to solve genetic problems. The genotype is referring to the genetic makeup of an individual. So in other words, what is their allele combination for that gene? There are 3 possible genotypes that a person can have, or 3 possible allele combinations. Without looking at the terminology, let's just think of the possible combinations. Well, they could have 2 dominant alleles, right? 2 big, I'm just going to use A's as an example. They could have inherited a dominant allele and a recessive allele from the other parent. Or they could have inherited 2 recessive alleles, one from each parent. If an individual inherited 2 dominant alleles for a gene, we call that condition homozygous dominant. Homozygous, homo means the same, right? And both of their alleles are the same and both of them are dominant. So that would be homozygous dominant. If they inherited one dominant allele and one recessive allele for a gene, then they are called heterozygous. And if they have two recessive alleles for a gene, they are called homozygous recessive. Okay, so those are the only three possible genotypes that can exist for a gene. Another term that I want to point out is this term carrier. And you probably have heard of this word before. For example, you might have heard somebody say that they are a carrier for a certain disease like cystic fibrosis or something like that. If someone is a carrier of a disease or a trait or something like that, that means that this individual carries the recessive allele for that disease or trait, but they don't express it. So somebody may carry the allele for cystic fibrosis, but they don't actually have cystic fibrosis because it's a recessive allele, and you need to have two of those recessive alleles to actually express the disease. So an individual that is a carrier must be heterozygous. And an important thing to remember about carriers is that they do not show signs of the disease, but because they carry the allele, that means that they can pass the gene on to their offspring. And so let's say like two parents are carriers of a disease. Both these parents don't actually have the disease, but if they give their child if each parent gives their child the recessive allele for that disease, then their child could actually inherit the disease and actually express the disease. The final term that I want to throw out there is this term phenotype. And this is the physical appearance of an individual based on their genetic makeup. So for example, do they have freckles or no freckles or so forth. This is easy to remember because the genotype of an individual is their genetic makeup, as the name implies. And the phenotype of an individual is their physical appearance. So PH, PH, they both start the same. Let's look at the image over here to try to apply some of the terms that we just discussed. So the first gene pair up here is a gene that can potentially cause cystic fibrosis. However, the dominant allele results in a healthy individual, and the recessive allele, if they inherit that would lead to a mutation that would cause the individual to have cystic fibrosis. So this individual, what is their genotype? Well, their genotype is homozygous dominant because they have two of the same alleles and both of them are dominant. What would their phenotype be? Well, they would be healthy, right? They would not have cystic fibrosis. Now let's look at the next gene. This is the gene for skin pigmentation. The dominant allele, if you have that variation of the gene, results in normal skin pigmentation, and the recessive allele, if you have that variation of the gene, would result in albinism. Well, this individual inherited a dominant allele from one parent and a recessive allele from the other parent. So what is their genotype? This individual would be heterozygous for skin pigmentation. And what would their phenotype be? Well, they would have normal skin pigmentation because, again, they have the dominant allele. Heterozygous individual always expresses the dominant allele. If we're looking here, let's look at the last gene. This is a gene for freckles. We can see that we know that this is something that would just be given to you, that the dominant allele causes freckles, the recessive allele does not cause freckles. So this individual has two recessive alleles, so what's their genotype? They are homozygous recessive. What's their phenotype? Well, their phenotype would be no freckles. They would not have freckles because freckles are caused by a dominant allele and they have two recessive alleles. Another important thing that I want to point out is that this individual here is a carrier for albinism because they carry the recessive allele and they do not express it. So this means that this individual could potentially pass on this allele for albinism to their offspring. Before we start solving some genetic problems, I want to introduce two important laws that describe the inheritance pattern of traits. The first law is known as the law of segregation, and you want to understand the definition of this law. This law states that alleles separate randomly during the formation of gametes so that each offspring only receives one allele for a gene from each parent. Which allele an offspring inherits is random, so therefore there's a 50% chance which allele the offspring will receive from a parent. Let's look at the image down here to illustrate the law of segregation. Here on the top, we have a parent germ cell. Remember, a germ cell is a diploid cell that divides to create either the sperm or the eggs. In other words, germ cells make gametes. Germ cells will always have 2 copies of a gene, and whenever this cell divides to make gametes, those copies of the genes will separate randomly into the gametes. So, for example, if the germ cell contains 2 different alleles for a gene, like this big D dominant allele and this recessive allele, the chance that a gamete will have the dominant allele is 50%. The chance that the gamete would have a recessive allele is 50% because they split randomly. The second law that's really important to understand in order to understand what controls the inheritance of genes is the law of independent assortment. This law states that the inheritance of one trait does not affect the inheritance of another trait. For example, the inheritance of eye color does not affect the inheritance of height and so forth. So most traits are inherited independently of each other. Now we're at the fun part where you're going to actually learn how to solve some simple genetic problems. The first set of genetic problems that we're going to solve together are caused by genes on the autosomal chromosomes. The second set of genetic problems that we're going to be looking at specifically look at the inheritance of genes found on the X chromosome. And the reason that we look at this separately is because genes found on the X chromosome affect males and females differently in their inheritance pattern. In order to solve any genetic problems, you're going to be using what is called a Punnett Square. A Punnett Square allows you to predict the probability that a certain genotype or phenotype will occur in the offspring. So let's look at an example together, and I'm going to give you an example of how you want to basically set up all of these problems. And if you follow this pattern each time, it's going to be really easy to solve any genetic problem because you're just doing the same steps over and over again. So let's look at this example. Cystic fibrosis is caused by an autosomal recessive allele. So it's already telling you that it's caused by a recessive allele. That information is going to be given to you. If both parents are healthy but are carriers of cystic fibrosis, then what is the probability their child will have cystic fibrosis? Okay, so we're trying to figure out if both parents are carriers of cystic fibrosis, what's the probability that their child would actually have cystic fibrosis? So first thing I would always do is make a little key with the alleles. And in this case, again, you always want the dominant allele to be a capital letter and the recessive allele to be a lowercase letter. And you want to use the same letter. So if you're going to, you can pick any letter, but if I pick F, I'm going to have a capital F and a lowercase f. So in this case, we know cystic fibrosis is caused by an autosomal recessive allele. So having the dominant allele would be the healthy allele. And then having the recessive allele would cause cystic fibrosis. So that's a cystic fibrosis allele. Next, I would figure out what's the genotype of the parents. It says both parents are healthy but are carriers of cystic fibrosis. We know that carriers of a disease means that they have to be heterozygous. So each parent has to have a dominant allele and a recessive allele. Okay, so that's what a carrier is. And so both parents are carriers. Step 3 is you're going to figure out, what's the probability that their offspring will inherit these allele combinations. Then the next thing you're going to do is you're going to write down the possible gametes that each parent can produce along the top of the square here and along the left side here. Okay, so you can pick either parent to start with. I always pick the first one that I wrote out. So what are the possible gametes that this individual can make? Well, remember, the alleles separate in the gametes, so they could have one gamete that has a big F, and then the other gamete would have a little f. So these are the possible gametes that parent can make, or the possible sperm if that was a male, or possible eggs if that was a female. Now let's look at the possible gametes the other parent can make. That would go along here. It's the same thing. Big F or little f is what they could give to their offspring. Now let's look at the possible fertilization events. Okay, so if this gamete and this gamete fertilize each other, then the offspring would be big F, big F. If this big F if this gamete and this one fertilize each other, the offspring would be big F, little f. Then we bring down to this square. If these two gametes fertilize each other, then it would be big F, little f again. I always, like if there are two different letters, I always put the capital letter first. You want to stay consistent when you do problems like this. And then in the last square here, if this gamete fertilized that gamete, it would be little f, little f. Okay, so what was the question again? It was, what's the probability of the child having cystic fibrosis? Well, since cystic fibrosis is caused by a recessive allele, we know that the only way that you could actually have a recessive trait is if you have two of the recessive alleles. Okay, so of these possible offspring, only one of the four would have cystic fibrosis. So what's the probability? 1 out of 4, or 25%. So there's a 25% chance that their offspring would have cystic fibrosis, just based on probability. Hopefully that makes sense. Next, you're going to try to do a very similar problem on your own, and then I'll work through it with you. So try this problem yourself, pause the video, and then I'm going to work through it with you to see if you were able to solve it. All right, now let's try it together. Cystic fibrosis is caused by an autosomal recessive allele. So this is the same as before. So I would always start out with my key. And in this case, I would say big F equals the healthy allele. And little f equals cystic fibrosis. If one parent is a carrier of cystic fibrosis and the other parent is homozygous dominant, then what is the probability their child would have cystic fibrosis? So first thing you did is you came up with your key. Next, you want to write out what the parents are. So one parent is a carrier. I know that a carrier is heterozygous always, and the other parent is homozygous dominant. So they're going to have two big Fs. Now it says, then what is the probability their child will have cystic fibrosis? So now I got to do my Punnett Square. That. I just write down the possible gametes this parent can make. That's a big F or a little f. Then this parent, big F or a big F. There's only one possible allele they could give their offspring. And then I combine the different possible gamete combinations in these squares. So this one would be big F, big F. This one would be big F, little f, big F, and big F, little f. So in this case, what is the probability their child will have cystic fibrosis? Well, in order for a child to have cystic fibrosis, they would have to have two recessive alleles. Is that possible with these two parents? Can they make a child with two recessive alleles? No. So 0 out of 4 of them, or in other words, 0% chance that their child would have cystic fibrosis in this case. So if one parent's a carrier but the other one is not, then there's no chance that their child would actually have cystic fibrosis. All right, here's another example. Try this example yourself as well. All right, let's work on it together. Huntington's disease is caused by an autosomal dominant allele. Right away, I would just stop and I would make my key. So I know it's caused by dominant allele this time, okay? So some diseases are actually caused by dominant alleles. Not all are caused by recessive allele. So here's Huntington's. It's caused by a dominant allele. So that means having a recessive allele, if you inherited that, that would result in you being healthy. So that's the healthy allele. If one parent is heterozygous for this gene, so I'm already going to pause there. I know one parent's heterozygous. I don't even read the whole thing sometimes because you want to take it step by step. So that parent's heterozygous. The other parent is homozygous recessive, so they have to have 2 little h's to be homozygous recessive. What is the probability their child will have Huntington's disease? All right, so I just draw out my Punnett Square again. So I'm just going to make it a little bigger. Boom, boom, boom. What gametes can this parent make? Well, big H or little h. 50% chance that the gamete would have a big H, 50% chance it would have a little h. This parent can only give little h's to their offspring, so we just separate them. And so there's a 100% chance that this parent was going to give their offspring a recessive allele, right? So let's see what are the fertilization events that can happen here. So this big H and this little h combine. This little h and that little h combine. This big H and that little h. And that little h and that little h. So the question now is asking, what is the probability their child will have Huntington's disease? Well, we know Huntington's disease is caused by the dominant allele. So that means that you only need one big H to have Huntington's disease. And we can see that that happens in this offspring here and this offspring here. So 2 out of the 4 possible offspring would have a dominant allele and have Huntington's disease. So you could express it as a fraction. So 2 out of 4 or 1 out of 2. Or you could express this as a percent. So that would be 50%. 50% chance that their offspring would have Huntington's disease. Or you could say 1 out of 2 of their children would have Huntington's disease. Once you're able to solve a simple Punnett square, then you can actually determine the probability that an offspring will have more than one trait just by setting up multiple Punnett squares and then multiplying their probabilities. And let me just give you an example of this. First, let's talk about the steps to solve a genetic problem when we're looking at more than one trait. First, for example, if we're looking at 2 different traits, such as cystic fibrosis and Huntington's disease, you're going to set up 2 separate Punnett squares, one for each disease or trait that you're looking at. You're going to then determine the probability that they will inherit each trait, that a child will inherit each of these traits. Then to figure out the probability that the child would inherit both of these traits or both of these diseases, you just multiply the results that you get. Let's just say that I'm looking at 2 diseases, cystic fibrosis and Huntington's disease. And I want to know, what is the probability that an offspring would inherit both cystic fibrosis and Huntington's disease from their parents? And I found out that there's a 25% chance that their child would inherit cystic fibrosis. I did a separate Punnett square to figure out that there was a 50% chance that they would inherit Huntington's disease. But I want to know, what is the probability that they will inherit cystic fibrosis and Huntington's disease at the same time? The way I would just simply figure that out is I would take the probability that I got in this Punnett square, which is 1/4. I like to work with the fractions in this case. You could work with the percentages as well. But there's a 1 out of 4 chance that they would have cystic fibrosis. What's the probability they would have Huntington's disease? Well, 2 out of 4 or 50% chance. And in order to figure out the probability that the child would have both cystic fibrosis and Huntington's disease, I would just multiply those probabilities there, those fractions. And so in order to multiply, this is where most students get this wrong, is they just forget how to multiply 2 fractions. When you're multiplying fractions, you want to multiply the numerators and then the denominators. Okay, so 1/4 times 2/4, to figure that out, I do 1 times 2, which is 2. And then I do 4 times 4, which is 16. So that would give me 2 out of 16. Or you can reduce that to 1/8. So there's a 1 out of 8 chance that their child would have both cystic fibrosis and Huntington's disease. If you express this as a percent, you just do 1 divided by 8, and you would get 0.125 on your calculator, and then you just multiply it by 100. That would be 12.5%. So you could write it either way, but there's a 1/8 chance, or you could express it as a percent, which would be a 12.5% chance that their child would have cystic fibrosis and Huntington's disease. The cool thing about this method is I could actually look at 3 different diseases and figure out the probability that a child would inherit all 3 of the diseases from their parents. So you can look at as many diseases as you want when you're doing Punnett squares this way. The last set of genetic problems that we're going to learn how to solve are those that involve X-linked traits. X-linked traits are traits that are caused by genes on the X chromosome that have nothing to do with gender. And this occurs because the X chromosome is really long, right, compared to the Y chromosome. The X chromosome is really long, and there's actually a lot of genes on there that have nothing to do with sex determination or anything like that. For example, some of the X-linked traits found on the X chromosome include color blindness, certain forms of muscular dystrophy, as well as hemophilia. So these are inherited on the X chromosome. An important thing to remember is that males are XY and females are XX. So this means that males only have 1 X chromosome, females have 2 X chromosome. And this is really important because having this different number of chromosomes in males and females changes their probability of having these different X-linked diseases. So X-linked traits show a different probability of being expressed in males and females. And this is because males will always express the recessive X-linked allele if they have it. Okay, so if they inherit a recessive X-linked allele from one of their parents, they'll always express that allele. Whereas females can inherit a recessive allele from one parent and then a dominant from the other parent. And so that dominant allele, X-linked allele, can mask the recessive allele. And you can see that here. So females can be carriers for X-linked traits, but males cannot. Let's look at an example involving muscular dystrophy, which is a muscular degenerative disease. This is actually caused by a recessive X-linked allele. So when you're writing out X-linked traits, you always want to include the X with it, okay? And then you want to write the little allele as a little superscript above it, okay? So in this case, muscular dystrophy allele is recessive, I said, so I would always write it X because that's signifying that this is a gene found on the X chromosome. And then I'd put a little N above it, signifying that this is caused by a recessive allele. And then that means that the unaffected allele or the healthy allele would be X with a dominant allele, so big N. All right. So what are the possible female genotypes that could exist? Well, female could be homozygous dominant, so she's big N on her X chromosome. She could have a big N or a little n, so she could be heterozygous, or she could be little n on her X chromosomes. So what are the possible male genotypes? So remember, a male is always going to be XY, and they inherit their X chromosomes from their moms. And so the male could inherit a dominant allele from his mom or a recessive allele. So he could be big NY or a little ny, but he can't be heterozygous. So if he inherits the recessive allele, he will always express it. He will always have the disorder. There's no way for him to inherit a dominant allele to mask the recessive allele. Whereas a female can be heterozygous. She can inherit a dominant and a recessive allele. When we start solving these problems, you're going to start seeing what effect this difference has on the probability of somebody's offspring having a certain X-linked trait. So let's look at this example together, and then I'll give you a couple examples to try on your own. Muscular dystrophy is caused by a recessive X-linked gene. I will always tell you whether it's an X-linked trait. So do not just assume that a trait is X-linked without me telling you that it is. So muscular dystrophy is caused by a recessive X-linked gene. And so you just set up a key just like we did before. So we know it's recessive and it's X-linked, so you need to have the X's drawn. It's recessive, so it's a little n. Muscular dystrophy, X big N is the unaffected one. If a mother who is a carrier for muscular dystrophy, so she's a carrier, she's got to be big N, little n. has a child with a man that does not have muscular dystrophy, well, we know since it's a man, he's got to be XY, and he does not have muscular dystrophy. So he's got to have the X big N for his X chromosome. Then what is the probability their son will have the disease? And what is the probability that their daughter would have the disease? Okay, so I'm always going to ask this question in the same way. I want you to be focusing on, like, if they had a son, what would be the probability that he would have the disease? And if they had a daughter, what would be the probability that she would have the disease? All right, so you set this up just like we did before. So if you're X big N and X little n, what gametes could this mother make? Well, those alleles would separate. So it'd be X big N or X little n she could give to her offspring. This male could give an X big N or a Y. And so let's combine these gametes together. If these two come together, we get X big N, X big N. These two come together, we get X big N, X little n. If this one and this one come together, we would get X big N, Y. And these two came together here, we would get X little n and Y. You're always going to want to answer the problems associated with X-linked traits in the way that I'm going to show you now, because I'm always going to ask you what's the probability their son would have the disease and what is the probability that their daughter would have the disease. So in this case, we know that the daughters are always going to be XX, so two of the squares are representing the daughters, and XY are representing the sons. So in this case, it's the bottom two squares. So what is the probability that their daughter would have muscular dystrophy? Well, if they had a daughter, the probability that she would have muscular dystrophy would be 0 out of 2. Because in order for their daughter to have muscular dystrophy, she would have to have 2 little n's. And there's no possibility in this case. What is the probability that their son would have muscular dystrophy? Well, if we're looking here, we can see that this one here, this son, would actually have muscular dystrophy. And that's because he has a recessive allele and he does not have a dominant allele to mask it, because that's not possible in males. So 1 out of 2 of the possible allele combinations for the son would result in muscular dystrophy. So if they had a son, there would be a 50% chance that he would have muscular dystrophy. But if they have a daughter, there's a 0% chance that she would have muscular dystrophy, just based on the fact that females are XX and males are XY. All right, so now I have 2 X-linked trait genetic problems for you to try on your own. Pause the video and try this, and then we'll work through it together. Now remember that you set up these X-linked problems very similarly to the other problems that we looked at. So let's work through this together. Red-green color blindness is an X-linked trait that is caused by a recessive allele. So I'm going to do my key like I did before. Since it's due to a recessive allele, I'm going to make a little r, and I'm picking whatever letter I want to represent the allele. So this would be color blindness. I always do a little key. It just helps to see everything than having to go back and read the problem again. So that means the dominant allele would be I'm going to just write normal eyes or normal vision. All right, so after you write the key, you got to figure out what the parents' genotypes are. If a female who is colorblind, so the mother is colorblind, so we know females are XX, so that must be her genotype. In order for her to be colorblind, since colorblindness is caused by recessive allele, she has to have 2 of them. So she is X little r, X little r. She mates with a male who has normal vision. So that's my cross. She's being crossed with a male with normal vision. So males are always XY. Well, what type of allele is linked to his X chromosome? Well, he has normal vision, so he's going to have a big R. And then it's asking what's the probability that their son would be colorblind if they had a son. What's the probability that their daughter would be colorblind? So you set this up just like before. We separate the alleles that the mother has, X little r, X little r. The father would be X big R. Y. And then set up your Punnett square. Like that. Bring these two gametes together in this square. So now we're figuring out what the possible combinations are in the offspring. Bring this one and this one together. I always, again, do the capital letter first no matter what. And then these two come together. X little r, Y. Bring these two together. X little r, Y. So let's figure out first, what is the probability that their son will be colorblind? Well, we're looking at the bottom two squares because this is showing the possible allele combinations in their son. Well, there's only one possible combination. It looks like both of these would be colorblind because they have the little r, right? So for son, the probability that their son would be colorblind if they had a son would be 2 out of 2, or 100% chance that he would be colorblind. Whereas for the daughter, let's put the daughter here. This is representing the allele combinations in the daughter on the top here. Well, in order for the daughter to be colorblind, because colorblindness is due to a recessive allele, she would have to have 2 little r's, okay? And there's no possibility for that. So there's no chance, 0 out of 2, that their daughter would be colorblind. So 0% chance. So if these two parents mated with each other, then if they had a daughter, there's a 0% chance she'd be colorblind. But if they had a son, there'd be a 100% chance that he'd be colorblind. And that's because if he inherits the recessive allele, he will always express it. Okay, there's no way for him to mask the recessive allele. Whereas even though the daughter inherits the recessive allele, she inherits the dominant allele from her father, so she won't show the recessive allele. And this further demonstrates why males tend to express or have X-linked traits more than females. So if there is a disease like hemophilia or muscular dystrophy or color blindness and so forth that are linked to the X chromosome, it's going to occur in males more frequently than females. This is the last genetic problem that we'll do together, so try this again on your own and then I will work through it with you. All right, so hemophilia is an X-linked trait that is caused by recessive allele. So you can pick whatever letter you want to use, but it's always easy to kind of think of something that relates to it. So H for hemophilia. So X big H would be the healthy allele. X little h would be the hemophilia allele. And if the mother is homozygous dominant for the hemophilia gene, so remember the mother has to be XX, so she's homozygous dominant. She's got 2 big H's. And the father has hemophilia, so he is XY. In order for him to have hemophilia, what has to be linked to the X chromosome? The little h. And the same thing again. What's the probability their son would have hemophilia? What's the probability their daughter would have hemophilia? So I just separate their alleles. So this is for the mother on the top of my square, my Punnett square. On the side, I'll do it for the father. And then I combine the letters or the gametes in the middle here to look at the possible offspring. X big H, X little h, from this combining with that. This one would be the same, X big H, X little h. This one would be X big H, Y. This one would be X big H, Y. So what is their probability their son would have hemophilia? Well, I'm looking at the bottom 2 squares here. Well, in order for the son to have hemophilia, he has to have a little h. None of them do, so 0 out of 2, or a 0% chance. What's the probability their daughter would have hemophilia? So this would be for the son down here. Now this is the daughter up here. In order for the daughter to have hemophilia, because she has 2 of the X chromosomes, she must have 2 of the recessive alleles. And none of these have that. So there's also a 0 out of 2, or 0% chance that the daughter would have hemophilia as well. So although the father has hemophilia, this is interesting. The father has hemophilia, the mother didn't, and there's no chance that the daughter would have hemophilia or the son would have hemophilia based on those parents. This is the end of the genetics and heredity chapter. If you have any questions, please comment below and I will try to address those questions.